Welcome to Lesson 6 of Module 3, Biological Diversity. In this video, we're going to be looking at evolutionary changes in specific populations, and we're going to look at how small microevolutionary changes can drive big evolutionary changes and speciation over time, and we're going to look at the specific examples of the horse and the platypus. Some meta-language that we're using today, macroevolution, so evolution that takes place over millions of years and results in new species arising. And then we have microevolution, which is the evolution that takes place over shorter periods and results in changes within populations, but it generally does not produce new species. Some new forms that arise within populations are sometimes referred to as varieties or races. Species, they're the largest group of individuals that can breed with one another, a gene pool that is isolated from gene pools of other species. And then we have speciation, which is the evolution of a new species involving the reproductive isolation of a group from the rest of its species with no genetic interchange. So there are some isolating mechanisms which we're going to refer to at the beginning of this video, which can prevent individuals from different species interbreeding. And these are the prezygotic mechanisms. These may prevent individuals from coming into contact, prevent mating when they do come into contact, or they can prevent fertilization if mating does occur. And this this actually stops any evolution between different species. So there's geographical isolation, and there's where species are separated by physical and geographical barriers such as oceans, deserts, mountain ranges, and glaciers. Ecological isolation, where populations occupy different niches within the same ecosystem. Temporal isolation, where the breeding cycles or active times of populations do not overlap. Behavioral, where behaviors such as mating calls and courtship rituals are different. Structural isolation, so the reproductive organs of different species are physically incompatible. Gamete mortality, where gametes fail to fuse in fertilization and a zygote does not form. And then there's sexual selection, where a selection pressure that also functions as a prezygotic isolating mechanism. And then some post-zygotic isolating mechanisms, and these are mechanisms that prevent a zygote of two different species from developing into a fertile adult. And this usually results from interbreeding between individuals from different species. So individuals that do result from this are called hybrids. So hybrid inviability is a mechanism of reproductive isolation, where the sperm fertilizes the egg of another species to form a hybrid. But this hybrid zygote has unmatched chromosomes, meaning it does not usually survive long. If it does survive and undergoes cell division, the offspring does not develop fully and will not reach adulthood. If there are hybrids that do develop into adulthood, they're usually sterile and incapable of producing offspring themselves. So when we refer to speciation, there are a few types. So allopatric speciation, and this involves geographic separation. So the spatial isolation prevents individuals of the separated subpopulations from interbreeding. So over time, these different environmental selection pressures and genetic drift can drive changes in the frequencies of different alleles of the two subpopulations. So eventually, these two subpopulations may diverge genetically and structurally, where they are considered no longer the same species, where they can no longer interbreed, and this is referred to as speciation. There's sympatric speciation, which occurs when the new species develops within the same geographic region as the parent species. And this is usually common in plants rather than in animals. And this can occur due to different microhabitats having different selection pressures, or polyploidy, where it's caused by abnormal cell division, resulting in a change of a number of the chromosome set. So here we can see the allopatric speciation. So we can see that these populations have been isolated by a physical barrier. So we can see that over time, that the different selection pressures of both the different environments actually cause the populations to become different. There is a process known as adaptive radiation, and this is the rapid divergent evolution of a large number of related species from a single common ancestor. And this is what we saw in the finches of the Galapagos Islands. So the organisms evolved different adaptations in response to the new conditions and opportunities available in the different niches that they occupied. So it can result in a wide diversity of species, each with unique adaptations to their environment. And we see that in the beaks of the finch, and that's based on the types of seeds, but also the other foods that they eat. There are some genetic factors which drive evolution. The first is genetic drift, and this is the change in the genetic makeup for a population due to chance rather than natural selection. The bottleneck effect, and this is where we see population size and genetic diversity significantly reduced by an event, often a natural disaster, and can lead to increases in interbreeding. And then there's the founder effect, where a small group of individuals breaks away from the main population and they then colonize a new habitat. So, this founding population usually has less genetic variation than the original population and experiences slightly different selection pressures. If we look at the evolution of the platypus in particular, it is a monotreme that has diverged from the main mammal lineage approximately 165 to 180 million years ago. It is an egg laying mammal. So we know that it lays soft, leathery eggs. It produces milk from pores in the skin, and they also have a venomous spur on their hind leg and a sophisticated bill that detects electrical impulses of prey animals. So it also has some primitive characteristics. So it retained the egg laying from its reptile ancestors, and it also has a reptile like cloaca. Genetic analysis shows the modification of the venom gene from reptiles. So we actually see that, and we refer to it as convergent evolution from snakes. The modern species, however, are smaller than their ancestors and more specialized for an aquatic lifestyle, and its distribution has become restricted to Australian rivers. And then we see the evolution of the horse. And this is well documented, much more documented than the platypus. And it's the most complete transitional series that we have in the fossil record. A transitional series is a group of fossils that shows microevolutionary change from one form to another. And we saw the changing environment select new traits through a series of slightly different species. The general trends included they are a greater size, proportionally larger brains, improved running ability via the loss of the outer toes, longer leg and toe bones, more developed hooves, and tougher teeth. We say that some of this evolutionary change, especially in the leg and the toe bones, was due to the changes in the geographical environment that meant that a foot with no outer toes and a longer leg meant that they could run actually better. Thank you for watching lesson 6. Make sure you tune in for lesson 7.